Hi everyone, my name is Joelle van der Sprong, and I'm currently doing my PhD at the Ludwig Maximilians University in Munich. And for my project, I worked on genomics to aid in species identification in sponges with special attention to Haplosclerids. So why is assessing sponge diversity so important? First of all, sponges are ubiquitous in almost all aquatic environments, wherein they fulfill a multitude of ecological functions, such as providing nutrients to higher trophic levels, or creating heterogeneity of reef ecosystems. Furthermore, they're known to produce bioactive compounds that could be of pharmaceutical and biotechnological interest. To understand species diversity in ecology, however, it's important to have unambiguous species identification and delimitation. So the taxon of interest here is the Haplosclerids. It's one of the most speciose orders with currently over 850 species described, and it is known to have the highest sponge species richness on tropical coral reef systems. Also, it comprises many species that produce bioactive compounds. However, this order is also a taxonomic conundrum. Almost every family within this order is polyphyletic. For example, the family Petrosidae, shown in red in the 18S phylogeny on the left, is scattered throughout the different clades. This is mostly the result of incongruence between morphological and molecular taxonomy. So when assessing species diversity in sponges, we encounter different problems. One of them is, for example, the lack of deterministic characters, which are also influenced by environmental conditions. Also, due to their simplistic nature, many sponges are considered to be cryptic species, meaning they're morphologically very identical, but represent genetic distinct entities. But also molecularly, we're not able to solve the taxonomic puzzle. Standard gene markers used for many metazoan species do not solve at species or population level for Haplosclerid sponges. Furthermore, there's often co-amplification of non-target organisms, and due to the bioactive compounds that they produce, there's often inhibition when processing them in the lab. Hence, to better understand species diversity, there's a high need of new multi-marker methods. For my project, we've designed a universal probe set for the Haplosclerids. Via collaboration with Grace McCormack, we had access to unpublished transcriptomic data of Haplosclerid sponges covering the 3 major clades. Based on this information and additional data we collected from literature and GenBank, we designed a multi-marker species delimitation assay, with as aim to capture the broad diversity across the order. The MSDA was designed using the workflow of phylogenetic. Using this workflow, we identified the loci that were shared between the transcriptomes. Eventually, we designed 137 different base sets that resulted in 20,000 unique baits of 80 base pairs long. Then we used target capturing to fish out the variability using these baits, and this information can then be used to re-infer phylogenetic trees or to do phylogenetic inference. Between 2012 and 2017, 172 Red Sea Haplosclerids have been captured or collected across the Red Sea area. This creates for us a new or unique opportunity to test our MSDA for both taxonomic and biogeographic resolution. During the second phase of my project, I'm also going to test this MSDA on type material from various museum collections. So based on the MSDA and the target capture approach, we were able to retrieve over 230,011 million reads for the specimen. The number of loci recovered for each specimen was on average over 600 loci. Based on this information, we created alignments that we concatenated from the retrieved loci, and we created a 50% complete data matrix, which means that on every locus, there's 50% of the total specimen occupancy. Now, currently, this tree is still being evaluated, but based on first observations, we do see reoccurring patterns that match our expectations. For example, in clade A, we see transcriptomes landing that, according to previous studies, should also be landing in this clade. Also, clade A is supposed to have many species belonging to the families Gallimidae and Polyspongidae, which is also something we found back in our results. The similar findings were found for clade B, wherein we also found back transcriptome data that were also used for bait design of 3 species that were supposed to land together. And the same accounts for grade C, where we found transcriptomes landing together from Haliclona indistincta and Haliclona viscosa. These specimens are known to look morphologically very identical to one another. We also found other specimens that still need a formal description, but look morphologically very similar, as you can see here in the in situ pictures below. And then we found a clade wherein specimens were morphologically recognized as the genus Xesospongia, and they land nicely together with one of the transcriptomes belonging to Xesospongia testudinaria. However, we would like to emphasize that many samples still await a formal morphological description. This is something I will be focusing on in the next few weeks, together with Nicole de Voogd, who is also a leading expert in Haplosclerida taxonomy. Another approach we're working on is the VPP species delimitation analysis. It has many advantages, but the main one is that it doesn't depend on an a priori defined species hypothesis and is suitable for multilocus datasets. Using this integrative approach, we hope to set a new baseline, as it can provide a more robust phylogeny. It can also lead to more meaningful taxonomic revisions and better interpretations of biological and ecological observations. And how can this work benefit future work in genomics? Well, for example, using sequence reference genomes and supplement this with genome skimming to study population structures and also dynamics using SNPs. And subsequently, this can aid in understanding evolutionary histories, resilience, and adaptation of species, but it can also improve conservation efforts, for example, by providing tools for biomonitoring. Well, this project wouldn't have been possible with the aid of many other people, so I would like to thank the technical assistants, the collaborators that provide us with knowledge and data, but also the projects involved in the sample collection. And of course, I would like to thank the DFG that provided funding for this project, and thank you all for listening to my presentation.